Broadcasting from Silicon Valley, California, this is Conversations with Jennylyn. In for Jennylyn today, I'm Adib Karam. Today we have a very special guest for you, Dr. Samer Hattar, and I'm saying it in an English way. I'll say it in an Arabic way because I know we're going to be getting some Arabic people watching this. Samer Hattar, Dr. Samer Hattar. He's the chief of the section of Light and Circadian Rhythms, part of the National Institute of Health, the NIH as we all know it. And Doctor, welcome to the program. Thank you for being here on Conversations with Jennylyn.
Thank you, Adib. Thank you very for the invitation.
All right. I want to talk a little bit about your history. You were born in Jordan. You went to some of the best schools in Jordan. Basically, the best school, Terra Santa. It's a Catholic school. You were born into an Orthodox family, Lebanese mom. And tell me, you came to the States for college, right? You did not go to college in Jordan, or did you go to college in Jordan?
So I went to college in Irbid, north of Jordan. I did my undergraduates there. Then I went to the American University of Beirut to try to go to medical school, but then I get accepted into PhD at the University of Houston. I packed my bag, came to the University of Houston, finished my PhD in biochemistry and biology, then went to Johns Hopkins to do a postdoc in neuroscience for 4 years. And then I stayed as a faculty member at Johns Hopkins, went through the ranks and got tenured at Johns Hopkins. And in 2017, moved with tenure to the National Institutes of Health, NIH, and specifically to the National Institute of Mental Health. And I want to make sure that I'm giving this interview in my personal capacity and I'm not representing the NIH today. It's important to mention that because my area of research is different than the area we're going to discuss today.
Excellent. Thank you. Thank you for straightening that out. I appreciate it. My understanding is you started out, you wanted to be a priest, right? Was that in high school?
Yeah, in high school, I was determined to be a priest, Catholic priest. And then I saw Mendel's experiment, and Mendel, as you know, was a monk. And
it's okay.
So what happened is that I thought, so maybe I could be a scientist and a priest. So that's how I decided to go and do science and hopefully then become a priest.
That's great.
Honestly,
a lot of people don't realize, but a lot of science was, a lot of Catholic priests were scientists.
Absolutely.
And it contributed quite a bit to science. So it's interesting that you would say that.
Yes, absolutely.
So I want to talk about what you're doing, but I also want to talk about, since you're in the NIH and or part of the NIH, and with all the things that are going on with COVID and the vaccine, I wanted to talk to you a little bit about that. We hear the same thing on the news. We kind of hear the radical side from both. Get the vaccine, you're an idiot if you don't take it, blah, blah, blah. No, don't take the vaccine. It's not really a vaccine. So let's talk. And you, just to be clear, your office or your area is close to the area that's working. Tell me how close you are to the guys that are working on the vaccine or the testing on the vaccine.
Yeah, actually, it's funny because the building we're working on is building 49. Building 35, which is close to building 49, where the people who actually crystallize the spike protein of the coronavirus live in that building. So I don't want to give some medical terms that people don't understand. Crystallize literally means they know the structure of the protein. These proteins are incredibly small. You can't even see them with electron microscopy very easily. You have to use very special electron microscopic measurement to see them, and not at the level of crystallization. And yet they crystallize this protein, so they know the structure, they know where this protein binds to the receptors. And that's why it allows them to have this exquisite details of building sequences and knowledge that can target that specific spike protein. And that's because basic science allowed us to do stuff like that. and crystallize proteins.
So let me, from a layman's point of view, when you say I crystallize something, that means I'm thinking of it, I turned it into a glass where I can break it into very, very tiny pieces and study every tiny piece. Is that what you mean by crystallization, or
I
did I
mean, not
misunderstand?
necessarily glass. I mean, not breaking it, but you're
Right.
absolutely right.
Just as an example.
Three-dimensional structure of that thing, and you throw X-ray through it, and then you could build at the level of the angstrom. Like literally, you could figure out where each molecule is close to the other. And so this is amazing. Now it allows us to see protein in their all glory as scientists. And we don't want to bother people with the details, but literally you could know the three-dimensional structure of the protein. And usually proteins bind to other proteins by the simplest way to explain it, the key and lock situation, that they fit very beautifully with each other's, and that's how they recognize each other's. So if they don't fit very well with each other's, then they cannot signal, they cannot talk to each other's. They have to have this fit. And we'll talk more about this later as well.
Okay. So they've crystallized this, and you've talked to the scientists that actually did that, right?
I mean, they are in the NIH. I didn't talk personally to them, but I attended seminars from them. They talk in the intranet at NIH, and they answer our questions every week. They talk to us, so we have the knowledge firsthand. Absolutely.
Okay.
I don't do the research, as I told you, but I've heard it from them firsthand.
Okay. So let me understand. So the vaccine, one of the biggest things about that you hear is it's not a vaccine, right? So talk to me about, and it's why people are saying it's not a vaccine.
So as we said, we don't want to go to the radicals of each side. So when people say it's not a vaccine, I think what they mean is that it's not a typical vaccine. So, in the past, the word vaccine, the first use of the vaccine was literally giving the patient or the subject a weakened version of the infectious agent. So they either heat inactivated, or they give you a weaker form, and that then the antibodies produced to that weaker form. So then when you get the stronger form, you're ready. It's like amassing army at your border before somebody attacks you versus no army and somebody attacking you. You could tell that it'll be very different if you have the army on the border first, because you have the protection first.
The army being the antibodies.
The army being the antibodies, and the invaders being the invaders, viruses, bacteria, fungi, whatever microorganisms can infect you. But what is really cool is that now we have a new type of vaccines. So now we still have these vaccines where you use viral vectors. So for example, the Johnson & Johnson is a typical viral vaccine. The AstraZeneca is a typical viral vaccine. So the Sinopharm, the one from China, the Sinovac, also is the Sputnik from Russia. All these are typical viral containing vaccines. But I think the ones that people say it's not a vaccine, it's the messenger RNA vaccines, which are the Pfizer and Moderna. Okay. And I'll be happy to explain more why this is interesting, and so the nice thing about this vaccine is, because we know the whole structure of the protein that is important for the virus to gain entry into our cells. Mm-hmm. Now we know the sequence of the protein that requires this virus to bind to our receptors in our body. So now this sequence of protein is usually encoded by messenger RNA. So if you now can inject that.
So hold on one second. Sorry. So talk a little bit about messenger RNA, just so where everybody's on the same page.
This is really, honestly, people always say biology is complicated, but this is the simplest thing in biology. There is the very basic idea in the cell. Every cell in your body has DNA. The DNA is your genetic material. You have all these sequences. We don't have to say the number. It's overwhelming, but they are found in every nucleus of your cell, of every cell in your body, apart from your red blood cells. But in general, every cell in your body, right? Okay. And this DNA has section on it, very small section, that encodes protein. But the DNA cannot go to protein directly. It needs a messenger RNA. It's a messenger. So what happens, the DNA by RNA polymerases produces messenger RNA that leaves the nucleus. Remember, the messenger RNA doesn't go back to the nucleus, it leaves the nucleus. So that's important for later, because some of the lies about it modifies your DNA is completely lies. It goes to the cytoplasm, where then the ribosomes will take that RNA and form a polypeptide, which then becomes a protein. So the sequence is very specific. Like if you give me a sequence, I could tell you in 2 seconds what is the sequence of the protein, because the coding is very specific. Every code gives a certain amino acid, and we have 20 amino acids that form our proteins. I know this is a lot, but I'm trying to...
No, it is a lot, but you know what? I want to break it down. I want to break it down so people can understand it, so I can understand it, so we can make a decision whether we're going to take a vaccine or not, which vaccine to take, right? So from what I heard from you so far, look, there are vaccines that are the old vaccines. If you want the old vaccine, you don't want this non-vaccine, there's a Johnson & Johnson you can take, right?
If you don't want the non-viral vaccine, there is Johnson & Johnson.
There's Johnson & Johnson. So not all the vaccines are the same. The vaccine that was created in India, there was a vaccine created in India. Was that an RNA as well, or was that a viral?
Understanding, and I'm not so sure, my understanding is that it is an AstraZeneca that is being synthesized in India. So it's the Oxford AstraZeneca vaccine.
Okay.
Which has done wonders to England. If you look at England, death rate is plummeted now because most people are vaccinated. Soccer fans are back in the stands in England because of the AstraZeneca, and that's your typical viral containing. So they have an adenovirus that is replication inefficient. They cannot replicate. They put the sequence of the coronavirus in that adenovirus. They inject you with that virus gains entry into your immune cells, produces antibody. So that's your typical vaccination that has been there for hundreds of years. If you want that, take the AstraZeneca, take the JJ, take the Sputnik, take the Sinovac, Sinopharm.
Okay. So now this is good. Honestly, I didn't know there was that the differences in the
one of the things that happened this time, and it really, I should be careful because I'm in the NIH. I wish that the public understands the reason we were able to succeed so much against the COVID is because all the scientists started working on this. I know people who've never, I thought about changing my lab to working to COVID for a second. When we put all the effort of all the scientists around the world, like the Chinese, the minute they got the sequence, they published it. The minute they published it, the people in the NIH who crystallized the spike protein looked at the sequence, they designed that Pfizer and the Pfizer vaccine and directly started producing the Pfizer vaccine. It was within a few hours, in a weekend. So because imagine if we put our head to diseases in general like this, like cancer or infectious diseases, I think that tells us something about the power when you get enough money and enough support to actually put your effort to do this stuff. I mean, the government invested a lot of money, gave private companies a lot of money to try to get these vaccines. And that is thanks to American people, taxpayers, that we were able to do that.
Yeah. Wow. Let me take us back a little bit to the DNA. So the DNA is in the cell. The DNA, there's the protein part of the DNA, there's the nucleus. So everything that's happening, it's happening inside of the cell that you talked about, or is...
Absolutely. In general, everything in the simplest way, again, we don't want to go into the complication. In the simplest way, let's take your eye. If you take your eye is made of different cells. You have the retina, that is the light sensitive part. It's made of different cells. It's made of cells that project to the brain. It's made of cells that detect light. Every cell of your retina, as different as they are, they have the same DNA material. And again, this is an oversimplification, but it's correct.
No, I got you. Yeah.
They have the same DNA material in their nucleus. Every cell in your body.
And the DNA tells the cell what it's supposed to do, its function. Yeah.
Literally, the DNA has all the coding information that a cell has to do. So then you could ask me the question, if all of us have the same DNA, why do we produce an eye, a nose, a mouth? Because different DNA in different cells are regulated by different regulators. So the eye produces proteins that are important for photoreception. The nose produces protein that are important for the smell. The ear produces protein that is important for hearing. So that's how, even though it's the same DNA, the regulation of the DNA, the breaking of the code is different between different cells.
Okay. So now the coronavirus comes in.
Yes.
The coronavirus comes in and it attacks the cell. Yeah?
Yes. So the coronavirus is an upper respiratory virus.
Okay.
And that's why it's called SARS. I don't like the way they named it, but that's not something we want to talk about.
Okay.
And so it goes to your lungs, and then it attacks your lung cells. And once it attacks your lung cells, you destroy your lung capacity to breathe. And that's usually
right. So talk to me about, because I've also heard that it makes your blood thicker. It makes it attacks different parts of your body, right? Or was that misinformation?
Not at all, actually, unfortunately. Okay. And that's why I don't want to get infected by this virus. Actually, the reason I encourage people to take any vaccine, because even if you imagine the vaccine have side effects, I could guarantee you the virus has 20-fold more side effects than the vaccine. Because as you said, the virus is not only restricted to the lungs. It's known. In fact, in the mental health, and I could say that because that's public knowledge, they are finding the virus in the brain. So it's because it binds to a general receptor. It's called ACE2 receptor, but it doesn't matter. It's binding to a general receptor that is found in all of our cells. Okay. So it's actually not restricted to the lung. It's mostly to the lungs, but higher incidences of blood clot are incredibly increased with the coronavirus. Actually, part of the reason death was decreased is somebody figured out that if they give corticosteroid, they decrease the death from clotting. So stuff like that.
What is that? What is that? I'm sorry. What is that? Is that what kind of drug is that? I don't know.
Corticosteroid is just cortisol. It's cortisol. Yeah. But again, but the most important thing is that it really binds to these receptors and gain entry into your cells. And once it gains entry into your cell, then it hijacks your cell and starts producing proteins of its own self and replicates. And once it replicates, your cell explodes and produces more viruses and then more load. And then it overwhelms your immune system, your inflammation, blah, blah, blah. And then, I mean, I'm not a doctor, so this is...
No. So I'm going to try to, again, I'm a layman. I don't understand a lot of this stuff. So I'm going to try to explain it back to you what I heard, right? So you have the cell, right? The virus comes in, there's a receptor on the cell, right? The virus attaches itself to the receptor, right? And then
not the virus itself, but the spike protein. But you're right. Yeah, it's part of the virus. It's
part of the virus attaches, right? And then it starts making its own protein, right?
That it injects its RNA. Remember, the coronavirus itself is an RNA virus, so it injects its RNA into the cell, and then uses the cell machinery to produce protein from its own RNA that help it build more of the virus particle, so it replicates. That's how. So it literally uses the cell as a replicating, like the yeast uses the bread to replicate.
Right, right. Or like if a fire uses,
wood, yes, wood in your house
as fuel. So our cells become fuel for that. All right, good. I'm getting it. Okay.
Really, you got it. It's amazing.
Okay. All right. So now, in order traditionally, the way we've been solving this problem and the way the Russians and the Chinese and everybody else solved that problem, except for Moderna and Pfizer, right? Or except for the NIH who gave their technology to Moderna and Pfizer, right? So traditionally, we've been giving a weaker or a non-lethal vaccine. So when
virus that is usually incompetent in replication and doesn't cause any diseases.
Right. So now when the corona comes to the cell has already seen the corona, right?
Perfect.
And it basically puts up the defenses and kills it.
Yeah, but correct, because it's not the lung cells that protects you. It's usually the white blood cells, the B cell and T cells, and it's the immune system. But your concept is correct because they saw the sequences from the weakened or viral vaccine, produced antibodies already that are already circulating into your body. So by the time the virus comes into your upper respiratory tract, the antibodies are attacking the virus. They don't need a week or 10 days for the viruses to be produced. By that time, the virus went to your lungs. Because your viruses are pre-produced, they can defend you much better. That's the concept.
Okay, so again, we'll do it kind of. I want to take it really piece by piece. So the cell has seen the virus or a replica of the virus. We're looking at the old way, and we're going to talk about the new way in a second. It's seen it. It sends say, "Hey, I need help." The body creates these T cells antibodies, and they're in our bloodstream, right? They're in our in our bloodstream for a while. So when the virus comes in, when the coronavirus comes in, it comes in and destroys it. That's the original way. Okay. So now we're saying, okay, that way is okay, but this virus is a very smart virus, is what I've been hearing, right? This is a very smart virus. It's changing. We've already seen, they say there's a California version, a Florida version, an England version. So the cell itself doesn't recognize the new versions, right? The new version can attack you with the old.
This is a fantastic question, and I'm going to answer it. So first of all, RNA viruses are known to mutate. And by mutate, it means that when they replicate, they are not very good at keeping their sequence intact. This helps them because this is the concept of evolution. When you mutate, you produce new phenotypes that could help you survive better. So the viruses that produce phenotypes that make them better at transmission or better at evading your immune response, by definition, they're going to succeed more in the environment. So they take over. That's why the new strains that are powerful take over. That's bad for us. That's good for the virus. They are not really smart. It's just they are replicating and producing huge numbers that by random luck, they produce all these beautiful strains that are powerful. Now, the good news is that the vaccines seem to produce antibodies that are better from the natural infection from the virus. And usually it seems that most of the vaccine are at least partially effective against the new strains till now. Now, I agree with you, and this is something that keeps us up at night, that if new strains come in and they are not protective from the vaccines, then we have to come up with the new way. And this is where the messenger RNA vaccines will be more beneficial, because these messenger RNA are really modular. You only need the sequence of RNA that is enclosed in a nanoparticle of lipid. So what happens is that you could change that sequence pretty easily. In fact, my lab orders RNA sequences all the time, and you could get them by mail. It's not that hard to synthesize nucleotide sequences right now. It's actually pretty easy.
Okay, so I know what lipids are. They're like fat. This is what lipids.
Yes, exactly. Yeah.
Talk to me a little bit about phenotype, because you mentioned that. I want to make sure people are listening.
I'm sorry. I said
no, that's all right. I just want to make sure people. What does that mean?
It's the correct is the way we say character, like blue eyes. Like it comes up with a different character. So, for example, the virus, let's say, becomes better, more efficient at being in the water particles to be transmitted, or better at binding to your upper respiratory tract, so it has a higher level of infection. So anything that enhances the virus binding to your receptor is going to increase your transmission rate. Okay. So stuff like that, changes in the amino acid sequence of the protein, can improve the capability of that virus.
Got it. Okay, so now let's talk about the new, or what some people are saying, it's not really a vaccine, but what you're saying, it's the next generation of vaccines, right? It's really the leap forward that we made. And talk about how it's different. Then we're going to talk about why it's better, because obviously you think it's better, but tell me the differences first.
So I want to be careful. I don't think it's better. It's better. I think every vaccine has its benefits. I think what I like personally, and that's a personal opinion about these messenger RNA vaccines, is that they are very modular and they are very easy to change and improve, because they really don't need to enclose into virus and activate. You just need the messenger RNA in the lipid particle. And one thing that people really confuse is that this messenger RNA technology is not new. The technology is not new. I mean, we have been studying messenger RNA for years as scientists. And messenger RNA injections have been used for treatment of cancer, for treatment of other diseases as well. So it has been used in the past. This is not just came out because of the COVID vaccine, but it worked really well for the COVID infection. So this is not a completely new technology. So you would ask me, why didn't it work before? Like, we've been knowing about that possibility for 20 years. Partially because the messenger RNA are very unstable. And if you put them in the body without the protection from the lipid, they produce a super immune response that is not very good. It causes inflammation. So the reason we were able to use them, because we figured out a way to decrease their immunogenicity by covering them with the lipid virus. We means the scientists, not us. I mean, I've never worked on the COVID. I want to make sure that people know that. But the nice thing about the messenger RNA is that once it goes into the cell and produces the protein, it's going to be degraded. So there's no way it's going to modify your DNA. It's very unstable. Messenger RNA are known to be unstable. That's one of the biggest problems when you work with messenger RNA. They are the hardest to work with because they are highly unstable.
Okay, so I got you all the way to the part where, it's covered with a lipid, and like, talk to me a little bit. You said, there's no way it's going to damage, or I'm not, walk me through that a little bit slower. I apologize if I'm being slow, but
No.
I want to make sure I understand it.
You're not actually being slow. This is because, so one thing that people confuse the messenger RNA is with gene therapy. Gene therapy, you use adenoviruses to literally modify the genetic material of the patient. So you have a virus that will incorporate into the DNA. So you change the DNA. So you have to use viruses that will produce DNA that will jump into the subject's DNA. But in the case of Pfizer and Moderna, these messenger RNA will not even go back to the nucleus. They cannot go back to the nucleus because they are mature messenger RNA. So if they are in the cytoplasm, the DNA is protected. They're never going to hop into your DNA. If they don't hop into your DNA, they're never going to modify your genetic material. So anybody who tells you that these viruses will modify your genetic material, either they are willfully lying or they just don't know the biology.
Okay. So you have the messenger RNA. It's going to go, it's inside the cell. It goes to the
cytoplasm.
Okay, so to create, when it goes there, when it goes to the cytoplasm, talk a little bit about that piece.
Okay, so like any messenger RNA, because they synthesize a messenger RNA that looks like the millions of messenger RNA that are found in each cell of ours.
Okay.
Now the ribosome will come and recognize this messenger RNA and produce a protein.
Okay.
And this protein will be the spike protein that is found in the intact virus. But now you only have the spike protein. You don't have anything else from the virus. You don't have the code, you don't have the genetic material, you don't have all the other genes that the virus has. You only have the spike protein. Now, because your body never seen the spike protein, now the spike protein will be recognized by the immune cells as foreign protein, and antibodies will be produced against it. And in fact, go ahead.
No. So even though your body produced it, the immune system will see it as a foreign object?
Absolutely. That's the beauty of the immune system. The immune system fails sometimes in some people, unfortunately, and produce autoimmune diseases that are antibodies that attack the self.
Yeah.
But during development, when you're in your mother's womb, your immune system is literally surveilling all the proteins and develops, I don't understand it, to be honest. This is even more complicated for me. Like, immunologists have to explain this to you, and I don't know if they can. It surveys the protein and it knows the self from foreign. So if it never seen the spike protein during the gestation period, during the development, then even if your cells produce it, it's going to be recognized as foreign, because that sequence is not going to be recognized as self. It's going to be recognized as foreign. The minute it's recognized as foreign, is actually presented to the immune system, and I'll be happy to explain it, but it's not going to benefit at all. And the immune system produce antibodies that recognize that specific protein. And because the messenger RNA are designed to the most important part of the spike protein that helps the virus bind the cell, they have been very effective, 95% effective at preventing infection from COVID. So 95% of the people who get these vaccines don't get infected with COVID, which is amazing. And that's why the numbers are plummeting in the US right now, because we're vaccinating and the death is plummeting. Like we had one running week death below 500 for the first time from March last year, because older people got vaccinated, and so then they are protected against COVID. It's not 100%. You're going to hear cases where somebody is vaccinated and infected and possibly go to severe and maybe die. You will hear cases of that, but it's the numbers that matter, the percentages. I mean, it's become so infrequent, such event becomes so infrequent that we could really live our life again.
Okay. So to kind of stick with it, so the bodies produce, we there's a way now, instead of injecting the virus, to have the body produce just part of the virus. So anytime so when the body sees that part of the virus, actually it already created an antibodies for it. So it says, hey, that has that part, so I'm going to go ahead and kill it. So it only recognizes a part of it and it kills all of it. Yeah.
Perfect.
All right.
Because the antibodies that these vaccines are producing, the messenger RNA vaccines and the other ones, but specifically the messenger RNA vaccines, are what is known as a neutralizing antibody. When they bind to the virus, they neutralize its ability to bind to the cells. So the virus doesn't bind to the cell, it has no ability to replicate. It's finished.
It's going to die. Got it. Okay, so let's talk about some of the other things that come. Now I completely understand it. Before I move on, let me ask you, what scares you about the virus? What is there? Because a lot, we're full of.
What doesn't scare me? I mean, first of all, the fact that this virus is random. I mean, it kills young people, old people. Yes, it kills more old people, but it killed a lot of young people. It killed healthy people. It killed, it's not like you cannot just expect what's going to happen to you. You may get it and be asymptomatic, or you may get it and get severe infection. That's really scary because you cannot predict what it's going to do to you, because we don't understand it yet as well. The second thing is the long-term effect. There are a lot of people who had very mild effects, but they are what is called the long haulers. And the third thing is this clotting.
Talk about this thing. Talk about the long-term effect, because I think that's important. I got it, let's say, and minor, didn't feel anything. For me, what would be the long-term effects if I had it? Right, I didn't get it.
So, I mean, I've seen this on 60 Minutes, a program I really trust, and I've read about it. It's pretty amazing. There are people, if I remember the number, comes between 8 to 10%. It's not a small number of patients who got infected with the COVID vaccine, mostly women, actually, the majority of women, that although they had mild or very little symptoms when they got the virus, 6 months later, they are tired, they seem to have muscle problems. There are other issues that are developing within them. And as I told you, some people in the mental health are finding that this virus is even being found in the brain. So I worry about the virus we don't understand, that can increase your clotting in the blood, that can cause non-specific muscle weaknesses. Why would I want to get infected with this virus when I know I have a safe vaccine that has been given to 100 million or more people, more than 100 million? And literally the side effects are a little bit of tiredness, a little bit of shivers, a little bit of muscle ache. Why would I want to get a virus that killed 600,000 Americans? Why would I want to do that? It just doesn't make sense to me at all.
What do you say to the people that, clearly you get, this is new. We didn't know what to do. So before they were telling us to put masks on, don't put masks on. You're safer without a mask. You're safer with a mask. And then, when you look at the death rate, even though the infection rate is high, the death rate was what? Within 2.5%, or I don't know what the exact, but it wasn't very high. They're saying, well, now I'm 95% safe. Before I was 95% safe.
See, this is
talk a little bit about that, because I don't think. Go ahead. Sorry.
Before you consider the long haulers, the one that have long-term effects, before you consider what it could do to you in the future, that's really dead. I mean, you talk about people who have severe infections, like my dad, even though he was double vaccinated, got severe infection, his lung is damaged. I don't know his lung is going to come back to normal level. So you have lung damage. You have all these effects. Yes, you don't die, but why do you want to risk a new virus that our bodies have never seen? And it has such a general spike protein that binds to all your cells. Why do you want to risk it when all you need to do is take a simple vaccine? Now, I agree with you. The rollout scientifically maybe wasn't the best. We were confused. It's a new virus. We did a lot of mistakes as scientists, but that's one thing also you have to understand about science. Problem is we have to get enough data to recorrect the course. And this was like an experiment while things are happening. I mean, we're struggling to figure out what's going on. As I told you, like if people didn't figure out the cortisol effect, maybe much more people would have died. Stuff like that. On the spot, we were discovering ways to better treat the people. People are trying to produce like the monoclonal antibodies. Maybe they'll have an effect. Maybe they want to have an effect, some therapeutics. So we're trying to do all this stuff at the same time. So yes, there were some confusion. There were some mistakes. But the most important thing for scientists to do is to admit these mistakes. The same way we discovered as scientists that JJ has a 1 in a million chance of producing some weird clotting in females. So for example, if you're an 18 to 48 female, that's my own opinion, I would take the Pfizer or Moderna. But again, it's a very low. If you get the JJ, I will also take the JJ. But stuff like that, I mean, the scientists are being so careful about the side effect that we could detect a 1 in a million case with JJ. That's how serious and how determined we are to be very open about any side effects that these viruses produce. Because if we lie about that, eventually people are going to know. And it'll be a disaster not only for this vaccine, for science in general. So scientists are not stupid to do something that is known to be uncovered. We're not making money like the tobacco industry where, we have to hide the data because we're selling cigarettes. As scientists, we only sell our reputation and we want the public to trust us. So we don't want to lie about anything. It doesn't help us.
So in the past, there's been mistakes, right? Now, I'm not talking about Corona in general. So, man tries to fix something, or a human being, I don't want to say man because that sounds sexist, but what us as human beings, we try to fix a problem and we make it worse. Ronald Reagan, I think one of his things was, the worst thing you can hear is, or something like to the effect that I'm from the government and I'm here to help, right? It's kind of, there's a lot of mistrust, there's a lot of mistrust out there. From your point of view as a scientist, is there anything about the vaccine that scares you? Is there anything that you don't feel that could go sideways, or you're like 100% sure? You know what I mean? Like because
I can tell you, I would tell you in science we're never 100% sure because there is no 100% in science. But I would say honestly, believe me, if a scientist would see something wrong, they would be all over the channels. All the scientists. Science Twitter will explode, will be talking about it 24/7. We don't, as scientists, one thing we do, we're very critical on each other's. Actually, we review each other's papers, we give each other's hard time because we want to be sure as much as we can be that everything we're doing is not dangerous and is not causing more problems. Right. Now, I agree with you, we could sometimes do something that could cause more problem, but in this situation, we already have the numbers for 6 months. Remember, we started giving these messenger RNA vaccines for the third phase trials in July. And we have all this data right now for 6 months. There was not a single time in history that a vaccine had a safety levels that what the Pfizer and Moderna and the JJ vaccines have in the 6 months. And then we discovered something new. If we didn't see any side effect within 6 months, most probably 99.9%, we're not going to see these side effects later on. So I am pretty confident. I'm so confident that once Pfizer was given for 12, I called my family in Jordan and all my nieces and nephews got the vaccine. If I had any doubt, I would not give it 12 years old that still have 80 years in front of them, a virus that I'm worried is going to mess them up in 10 years. I could take it at 50 years old. If 10 years happen, I'm 60, who cares? But I wouldn't give it to a 12 years old. But because I'm so confident, because the science is so clear, messenger RNA is so well understood, the safety data is remarkable, the efficacy is remarkable, and most importantly, the COVID virus is nasty, is really nasty. Yeah. And to me, if you weigh the two, it's a very simple decision. It's a very simple decision.
Okay. Let me ask you about the vaccine expiring. We've heard stories about certain countries, letting, I know that over here, any vaccines, they've thrown away quite a bit of vaccines. If somebody doesn't make their appointment and they have extra at the end of the day, they don't put it back in the freezer, they throw it away. But there's other.
Sorry, I told you this vaccine, the messenger RNA is unstable. They have to be kept at very cold temperature, otherwise they get degraded. Mm-hmm. And it's sad that when they don't either, put them in good conditions, or the people who make the appointment don't show up, you lose these vaccines. And it's sad because people in India, they will do anything to get a vaccine right now, considering what's happening in India. So sometimes I feel like we're so privileged in this country that we say, oh, we don't want the vaccine, but people in India are dying to take the vaccine. You could see what this virus can do when it goes into this rampage of infecting a huge number of people. So
I'm sorry, I was going to ask you, okay, if you're in a foreign country, are you better off, or that you don't feel that they kept good track of their vaccine? Are you better taking a JJ rather than a Pfizer, because a JJ virus is not an RNA virus? Oh, I'm sorry, a JJ. I keep saying virus. Vaccine is not an RNA vaccine, or do those vaccines, are they restricted like that as well?
That's actually a really interesting question. I have to think about that, because if the JJ is, the beautiful thing about JJ, it needs only 4 degrees. And I guess if somebody really mistreated the Pfizer vaccine, you could be getting just water and thinking you're vaccinated, but you're not going to be vaccinated. You would hope that people would follow the rules. But I agree with you, that's a problem, because the Pfizer requires incredibly cold temperature. And I don't know, like if the electricity goes away. So I want to be careful about how I answer this. I can't give you a very clear answer. I would say in the US, whatever vaccine comes your way, I would take it.
Right. No. In the US, it makes sense that you would take the Pfizer or the Moderna.
But the JJ, honestly, any vaccine that comes your way, I would take.
And I get that. And I think there's more and more vaccines coming now that a lot of people have been vaccinated. But I was just curious, like if you were in a third world country, you're better off.
You would hope that they will, but it's a very interesting point. You would hope that they will have the resources to keep it, to transport it at the right temperature, to keep it at the right temperature. I mean, I know from my lab that if you keep these messenger RNA or these nucleotide sequences as wrong temperature, they won't work in the reaction because they get degraded. So that's interesting. I guess one thing maybe, but that will cost money. So is to do an antibody test after you get the second vaccine and see if your antibody titer is high. So that's,
you actually went into my next question. Is there a way after you've been vaccinated? Do you know that you're vaccinated?
Absolutely. There is a very easy way you could detect the antibodies themselves. So you could get the spike protein in a dish called ELISA. It doesn't matter what it stands for. And you just add your blood. And if you have antibodies, you could calculate how much. The reason I know about this, because I know people who have immune problems. They always want to do the antibody test to make sure that their immune system was producing enough antibodies after the injections. And in fact, most of them are producing antibodies, which is really great news for them. So you could do this, but that will cost money too. So that's why I said, that maybe, again, from our privileged situation, we could suggest that, but that may not be an easy thing for people to do as well.
Okay. I know you're not speaking for the NIH or really the agency since this is not your area of expertise, but how young do you think they're going to go? I'm not asking you to make a recommendation. I'm just thinking, how young do you think they'll go with kids with this vaccination as a recommendation?
My feeling is that if they test it on enough people and the safety record stays the same, they'll go as young as they can, and that is no problem. If the safety record stays the same. I mean, again, if you think about it, right now we have from Pfizer, I think from Israel, you have millions, from the US, you have 120 millions, some of them for 6 months that have been followed. And literally, you didn't hear of any severe side effects. I mean, literally zero. And that's a remarkable safety. So,
With a disease like this, it's big when you know people that have died from it. I know people that have died from it. I personally know people died from it, and I think somebody actually died after they've taken the vaccine. So it's important to know that once you've taken the vaccine, you're not fully vaccinated. And you said it was 2 weeks after the second shot or 3 weeks. How
four
long does it
weeks
take?
usually is after the second shot is when you have the full protection. And again, even after the full protection, I'm sorry, I have to be honest as a scientist, you still have a 5% chance. It has a 95% protection, but there are 5% of the people that still get infected.
So let's talk about that 5% because a lot of people get confused with that 5%. That's 5% of you catching it. Before, you pretty much, if you were
100%, yeah, sure.
you were at 100%, right? The 5%, right? So
no, so it's a low percentage, and you think about the low percentage of, and then even when you catch it, there is a very low chance of you going to even mild symptoms, let alone severe symptoms, let alone death. Okay. So no, I'm totally. I mean, the numbers are remarkable, but so I just want to say it's not 100%. It's not. Yeah, it's not.
No, but let's walk through this. Without the vaccine, if you're in an enclosed area where somebody has COVID, right? There's 100% chance that you're going to catch it.
Almost. There is almost
100% chance, right?
Depending on how long.
If you're in an enclosed area and you're vaccinated, there's a 5% of chance of you catching it, right?
And the 5% doesn't mean that if you stay like certain amount, you may catch it and just you kill it directly. So it could be 0%,
Yeah, but let's stick with 5. So we went from 100, if you're not vaccinated, to 5. Let's say now that you're at 5%, there's a 5% chance that you might die, right? Or no? Now that you're vaccinated,
it's much less. Even so much less.
Okay.
Yeah.
So now. Do we know anybody that's died after the 2 weeks of the second shot yet? I know as of 2 weeks ago,
I read one article in the New York Times that an older facility had a worker that wasn't vaccinated, and all of the old people there were vaccinated, and several contacted it. Only one went to severe and death. So we know, but again, the numbers are ridiculously small. That's why in England, like now, you have 2 to 3 people dying. It's just amazing. In Israel, you had a couple of days where it was 0 people dying. So it's just amazing. You could look at the US numbers now, and they are nosediving, the death, especially because we're vaccinating mostly the older population. If you look at the whole numbers, like even in California, there were moments where people would say they are following, but then they will find out that there were parties or a lot of people going to, so yes, there were mandates, but were the people following them? I mean, the best example I give is New Zealand, South Korea, China, these people who wear masks, Taiwan, the whole pandemic, they lost so few people because Taiwan get hit by the first SARS. So they were so scared of the second SARS. And so they knew, and they protected, they used the mask. I don't know why people worry about, the masks are so effective. Even, if everybody uses the mask, they are very effective. They help Taiwan and China maintain the numbers at low levels. So I think there are a lot of things that go into what happens in an open environment. As you said, like, people say they mandate and then they go have dinners without masks. So if the people who did the laws are not following them, imagine the people, like I've seen people who say, oh, I'm very careful, but I had guests yesterday and they said they've never seen anybody. I said, they said, but you don't know.
Yeah.
So people will make these assumptions. So it's hard. You have to do that.
I guess you go to like I get what you're saying, but you go to a state where I know people that have actually left California and have gone to Florida because they believe that the government doesn't know what they're doing. They're making you do things that
But
are
remember,
unnecessary.
it's not the government, it's actually the scientists. So I could tell you it's not the government. I mean, that's one thing I want to make sure people understand. Like in science, in the National Institute of Health, we're a government agency, but all our science is actually reviewed by scientists. Like I am actually getting reviewed in June 1st by scientists from University of Texas, from wherever, from all over the place, from private universities. So it's not true that it's scientists who do this. It's not
No, but
politicians.
what I meant by government, they say, okay, we're going to close down the restaurants. We're going to, make sure
Because
they
the
only eat
scientists,
outside.
yeah, because the scientists find out based on the data that they got, like look what happened in India, for example, that once you get a certain level of infective rate in the society, it becomes very dangerous for people to gather together, especially in enclosed areas.
Right.
So they suggest we need to cut this down or mask mandate. Now, this only would work if people comply. It's like,
No, I guess, yeah.
that's why
But
Asian countries like Taiwan and China, because people complied, it worked. But if they don't comply, it's going to take a long time to work.
so
And we lost 600,000 people. So that tells you we didn't do very well. We
Yeah.
didn't do very well. We didn't, like compared to Taiwan, I don't think they reached 1,000. The last I've and New Zealand. I mean, New Zealand is protected, and Taiwan, they are islands. But China, even China didn't, after the first initial part, I mean, they have been pretty much at the low end of infectivity because everybody uses masks, they measure temperatures. I mean, we don't want to become
they lock
so,
people in their house there, don't
yeah, we don't want to be so oppressive. But
Yeah.
what I'm saying is that it is scientifically, there is no doubt that lockdowns and masks have saved so many lives. There's just simply no doubt. You saw it in Brazil when they thought that they will get enough infection that they'll get herd immunity, and then they got new strains that started the same round of death. And Brazil was a mess for a while. You've seen it in India. You've seen it in Florida, actually. There was a point in Florida that it was a mess. You've seen it in California, that even though there was a mandate, people weren't being careful, especially in crowded areas. And there was a huge number of infections. You've seen it in New York. So I don't think it's blue or red or whatever. I think this is simply scientific. This virus requires particle from your mouth. If you protect your mouth, it's not going to be transmitted. It's like, sorry to say that, like the AIDS virus with sex. If you don't have, if you use a condom, you don't get AIDS. I mean, that's something we knew for the whole duration of HIV infection. So it's the same thing, but the mask, it just protects you because that's the mode of transmission. Now, I do agree with you. Why at the beginning they said mask may not help? Honestly, beats me. But I mean, there wasn't enough mask for the health workers, and they didn't want people. But I think you should have been more, because I don't like even simple lies from scientists. I don't like them because they produce the doubt that you're talking about. And we have to be careful because people don't understand the details like us. We have to be careful about what we say, and we have to be 100% honest. And that's why I love what the JJ did. They were very honest. They put the numbers. Everything is out there, and they said, that's the numbers, but this is what the vaccine does. It protects you from this nasty virus. The benefits outweighs the risks. And that's what we do in science. We always measure benefits and risk. We cannot be 100% sure about anything in life, unfortunately.
Thank you, Simon. I want to just do a quick recap on what we talked about today and what we learned. And I want to ask you also about what you're working on these days and what we should expect from your lab. Just to kind of recap, so this vaccine will go in to your body. it will send a message inside your cell to create a protein that the body will not recognize, and the body will create an
Not
antibody.
recognized as self, was not recognized
Yeah,
as
will not recognize as part of your body. So it will create antibodies, just to make it simple, to kill that, and it will stay in your system. But we don't know how long it's going to stay in your system, right?
That's a great question. So
Okay.
right now, people have been tested up to 6 months and still there, but we
Okay.
don't know if you need booster or not. But all you need possibly is one injection every year, like the flu virus. Maybe not, maybe yes. We'll see. I mean,
We'll see
this
what
is
happens.
something we have to see what happens.
And that's the way to do it. And we're saying this is the new kind of and it's absolutely no good if they didn't keep it at a certain temperature. You're basically just getting water. So make sure you're getting it at a reputable place, someplace that you trust. Otherwise,
I mean, honestly, in the US, I would be surprised if anybody would be that stupid to keep it at temperature.
well, yeah, and honestly, this show is on the internet as well as out of Silicon Valley. So people around the world might be seeing this. I want them to kind of get that information. And the last thing is that there's a way to test. If you want to spend the money, there's a way to go in and see if you have the antibodies. If you know, just to make sure you got it 2 weeks later, do I have the antibodies? You can go and request that test, pay for it if you want to.
If you want to be completely happy, tell them I want a quantitative ELISA. So they'll tell you actually how much antibodies, like they'll give you a quantitative number, how strong your antibody response.
Quantitative ELISA. Okay. So we don't know what that means, but that's what you should ask for, right? For us, for me, I'm sure you know what it means, but okay. Let me ask you about your work right now. Let me ask you about what you're doing, what should we expect what's exciting you right now,
So the biggest excitement that is happening in my lab, and again, I'm not allowed to talk about unpublished data, but so I'll talk about published data, is that we have started to uncover that although light is important for vision and your body clock, which is for vision, if you don't have light as a person who do interviews all the time, then everything is a disaster, right? If the lighting is wrong, everything. So vision, you understand. Biological clock, if you don't have light, you start free running and your day becomes night. You start having jet lags in your own town because you're sleeping up at the wrong time, waking up at the wrong time. It's a disaster. You have cardiovascular disease. But what we didn't appreciate is that light has direct effect on us as well. We kind of knew about it, but not in details. Like we knew that people who live in the Northern Hemisphere, in the winter month, they develop seasonal affective form of major depressions, a major depression that happens in the seasons. And now we're starting to understand, and that's all I could say, the circuits of how this light interacts with the body to actually affect mood-related behaviors in animals at least, and hopefully we're going to do it in humans. So I'm really excited because figuring out these circuits may help us to just simply use light to just enhance mood and sleep in people, just changing the light environment, which has a huge impact on neuropsychological diseases. So I'm very excited about that. That's the extent I could talk about right now.
That's awesome. I'm really looking forward to seeing what you're going to publish.
We're writing the paper, so hopefully it'll be soon.
So I'm hoping maybe after that you can come back and talk about that on this show.
I would love that. I would talk with much more confidence, and I could tell you numbers and details because the immune system is something I was very excited about, and I thought about moving to, but I'm not the expert. But this is my expertise.
Dr. Hattar, thank you so much for your time. I really do appreciate it.
Thank you so much.
Have a great day.
Thank you. Bye. Bye.
Broadcasting from Silicon Valley, California, this is Conversations with Jennylyn.